I stand on it. It goes without saying that had to have been a supreme confidence builder. Even in your already very successful and advanced career, that had to be a supreme confidence builder. But and as I said, we could talk about COVID for like the
Oh,
next 3 hours.
yeah.
But I wanna kind of shift back to like what makes Dr. Tal Zaks who he is today, and kind of keeps him moving forward. So taking sort of that
Hmm.
specific experience off the table, what other inflection points during your, CXO, VP, even research experiences with your previous companies have, in a similar but maybe more muted, more muted to the public way, influenced your confidence in your career trajectory? Your decisions to go on and be a board guy, be a founder, be the leader of Exsilio, for instance. Now,
Uh,
pretty big
it's a
open-ended question.
yeah, it's a combination of factors. It starts with the absolute joy of seeing clinical benefit come at the other end. I'll tell you a story. Back when I was at Sanofi, we had a drug that was in phase one, just the first phase of clinical research, and it targets something called CD38 for patients with a type of blood cancer called multiple myeloma. And this phase one had been going on for very long because we started at very low doses. We were going very, very slowly, and it wasn't clear that it was going to work. So people weren't excited to enroll. And one day the team comes back and tells me, you remember that 70-year-old patient that we had, and he just gotten a couple of doses. Well, he decided to go off protocol. He's no longer participating. And I'm like, why? We just lost a data point. This is hard enough as it is. And they say, well, here's what happened. We gave him 2 doses or 3 doses of the drug. He went into a complete response. He's 70-year-old. He's been feeling like crap for the last decade of his life. He finally got the opportunity to go to Europe and take the summer off and do that trip he's been dreaming of. So he decided he's not coming for his next dose. He's going off to Europe to live. Right. And
Live
then
his
you
life.
go,
That's
okay,
amazing.
I
I
guess
guess
I guess that's okay.
that's what we strive for, right? Improve quality of life.
Exactly. So,
Wow.
so it's those stories that you sort of that you live for. On the, I think I've been fortunate to have mentors and colleagues to be able to talk to and have sort of steered me right. Whether it's people like Steve Rosenberg, who is still there at the NCI now 50 years as the towering modern giant father of immuno-oncology. A lot of therapies we see today, the T cell therapies, all started in his lab back in the day. Um, I've been fortunate to have a supportive wife who understands what I do, and more than that, will actually tell me to my face when I'm talking nonsense, um, and tell me the stuff I don't want to hear. And I've been blessed with having colleagues. One of the privileges that we have in the world, in this ecosystem, is that the end game is something tangible that is easy to be passionate about. We're not making widgets. We're making people feel better, live longer. We're trying to cure diseases. We're trying to prevent horrible illnesses. It is easy to get that as a rallying cry. What's hard is then to figure out, okay, with all the unknowns in biology along the way, how do you do that? Now, I think in this phase of my career, one of the things I learned at Moderna, sort of through doing, is the importance of aligning return on investment with kind of innovation and doing good for patients. And it sounds trite, but it's actually not obvious, and it's not trivial to get right. The Our ecosystem has been so productive, but you have to understand the drivers of this ecosystem. Because if you look at other economic models of trying to translate innovation into healthcare benefits, they have not worked nearly as well. And so I judge it by the results, and I am deeply respectful of the underlying economic ecosystem that rewards innovation and rewards investment in high risk areas with a return on that investment. I was kindly invited to a recent summit that one of the National Academy of Sciences, Medicine and Engineering had about how to better align investing in innovation and therapeutics with unmet need. And one of my points to them was, I'm not ashamed of saying return on investment. When we talk about drug pricing, we often talk about kind of reimbursement. And reimbursement is a term that is fit for when I go out and have a meal and I need somebody to pay for the meal. We don't work for reimbursement. Let's be fair. We work for a return on the investment. My shareholders, my limited partners right now in venture, a lot of them are pension funds. They give me a pot of money. 7 years later, I need to give them a bigger pot of money back because they got to go pay those pensions. That's what propels our system. That is commercial investment. And for me, since I was fortunate to never actually do anything strictly for money, I became super curious to understand and immerse myself in an ecosystem where that part of the value is actually a very objective and legitimate yardstick. Sweat equity and capital equity tend to be rewarded differently. I've sort of lived now through both. And when investors at Moderna used to ask me, well, what's the stock price? Should I invest? Is it too high? Is it too low? What's it going to? I used to look at them and say, look, you can do what you want with your money. I'm actually investing something much more valuable than money at Moderna, and that's my time on Earth. Mm-hmm. So you be the judge of your money. I think sweat equity and capital equity are different. And these days, I'm curious to immerse myself in this side of the business of biotech, if I may, to understand what that return on investment looks like and how do I best align it with the things that I still inherently care about, which is translating science into medicine.
Yeah. Yeah. In that vein, I mean, one of the first questions that investors ask, one of the first questions that regulatory agencies ask when they're looking at investigational therapeutics is about unmet medical needs. What does it address? Lots of research and biotech builders thus go about their business searching for solutions to unmet medical needs. Why not? It potentially wins the favor of markets and agencies. But you, on the other hand, have shared a somewhat, at face value, perhaps contrarian opinion that unmet medical need is not really what drives innovation in this industry. So I guess I'd ask you for some explanation of that philosophy.
Explain yourself.
Yeah. Well, it struck me as I was starting out at GSK. I remember at one point we said, okay, we're going to focus on lung cancer because it's an unmet need. And being the flippant young guy that I was, I told the head of the division that actually if he wanted to have an impact on lung cancer, he didn't need to do much work. GSK already had the best drug in their portfolio at the time. And he goes, yeah, what is it? I said, it's Nicorette. Unmet need hasn't changed. And so we've had cancer for eons. And so it's not unmet need per se that's driven all the new medicines we have. So it's actually new understandings in science and technology that are driven primarily by curiosity. When Caitlin Carrico, whom I have a ton of respect for, started fiddling around with the structure of RNA to understand why in some cases was it killing cells and what could she do to the chemistry to stop it from killing cells and get protein expressed, she came upon a really interesting discovery, but nobody knew what to do with it. In fact, the IP lay fallow for years. They kind of licensed it to somebody else. And it wasn't until people who actually said, there's something that maybe we can get translated. And even then, the first folks who picked that up, their idea of what to do with it was just following their curiosity. Oh, can I reprogram a stem cell to go be something else? Which is super interesting scientifically, but we still don't know how to make a medicine out of it. And so for me, that bit of translation in the middle, pure innovation opens up the potential. That's the curiosity driven. That's basic science. That's basic technology. When Bob Langer discovered that he could make lipid nanoparticles, and Bob is a legendary MIT professor who's the father of the modern lipid nanoparticle, and frankly, one of the fathers of the current ecosystem as we know it of Boston Biotech. And that's because of his deep understanding that when he discovered he could do something, and that something was everybody else thought he couldn't do it and shouldn't be possible, that opened up a wonderful world of intellectual property. And and his understanding of the value of intellectual property and the ability then to showcase what you could do with it to attract investment, that's what started the ball rolling. And frankly, Moderna is sort of the culmination of the curiosity of people like Bob Langer, like Caitlin Carrico, but actually putting it together in something that now is useful. And in fact, even in the early days of Moderna, we were exploring a whole bunch of different things and trying to make different types of therapies with it. Moderna still is exploring the next frontier, whether it's cystic fibrosis or rare disease. Now we know vaccines work. And what happens is unmet need becomes the prism by which you focus the light. It's not the source of the light. The source of the energy is actually that scientific and technical innovation. And that's brilliant people who are just super curious to go show something or investigate something. But the ability to then focus that potential through the prism of unmet need is what gets us medicine.
I think that's a really interesting distinction, especially given where we are right now, I think with mRNA and you're positioning yourself right at the head of a brand new mRNA company, right? So, where there's still so much unknowns right around the science of mRNA and how it works and the delivery of it and how it's going to perform in different cell types and how we get it to express longer. It's such, there's so many different scientific questions. And we also, tend to, I think, celebrate mRNA at face value for the ability to manufacture more efficiently, right? You've talked about this in comparison to other cell-based medicines, right? So, I'd be curious to get your sense on the trajectory of innovation in this space, what you really think are some of the pieces here that are really driving further innovation in that space, right? And that we are starting to funnel into unmet need to some extent to make these into therapies. But what are those, what's stoking the innovation in the mRNA space today that has you the most excited, and what's at the root of that?
So I think the two current frontiers for mRNA medicines have to do still with the original questions in a way, except we've moved the ball down the field. The first is, what cell can you get the mRNA into to mediate the protein translation that would lead to a therapeutic benefit? Vaccines were the low-hanging fruit because all you need is a needle in the arm and it goes to the local lymph nodes, and all you need is a smidgen of protein produced and you mimic a viral infection and off you go. The next place where you can put it is likely intravenous, and I think that's also been proven. We've demonstrated, at Moderna back in the day, shown that we can inject mRNA encoding just for an antibody intravenously, and it goes to the liver and it will make you antibody and you get therapeutic doses of antibody. So can you get it to the lungs? Can you get it to the bone marrow? Can you get lipid nanoparticles into immune cells? Can you get into the brain? I think that's those are some of the frontier areas where people are trying to push the boundaries. Mm-hmm. The next, the other dimension is, well, what actually can you encode in the mRNA? And initially, the place you start is the stuff that's best understood because you want to minimize risk, and nothing is better understood than an antigen for a virus. And, it's no mistake that all the COVID vaccines all use the exact same spike protein that everybody now, knows what a spike protein is. Okay, that was easy. But you could actually make proteins that are much more complicated. You can make proteins that require several subunits to come together. And you can make proteins that have not been seen in nature. You can invent new proteins that have novel functions or improve on functions in very creative ways. I am curious about that frontier personally. That's where Exsilio comes in, because for me, Exsilio, this gene therapy company that I've now taken on, is actually at the forefront. It can combine innovation that comes from two different sources. One of them is mRNA and a lipid nanoparticle as a drug format. We can give it, it can go to cells where we understand it can already go to, whether it's the liver or immune cells, and it can translate protein. And you can actually give it repeatedly. You can give it once, twice, three times. In fact, Moderna and their lead indications have gone now for about a year in their first two diseases, giving it every 2 to 3 weeks, no problem. But the second thing is gene therapy. Can I actually integrate a novel piece of genetic material in a safe way into the human genome to produce a therapeutic protein? And then I don't have to keep treating that patient. Well, that's as close to a cure as I know. And so what this company that I'm now running is trying to do is basically take a page of evolution as a starting point of some elements that can actually do this in nature, but use modern tools of protein engineering to improve upon their utility so that we can actually use mRNA and a lipid nanoparticle as a way to bring gene therapy to the forefront, and do it not just by doing one or two small alterations of a genome, but actually to insert an entire piece of missing genetic material and cure a kid with a genetic disease, for example.
And this, my friend, is where the cliffhanger comes in, because we do have a hard stop. And because my next series of questions, we certainly cannot tackle in 4 minutes.
We cannot.
Those questions,
sadly.
Those questions being, if curiosity is to be rewarded, it must be funded. Who is funding curiosity?
Exactly,
Who's
right?
willing to fund? Who's
willing to fund these levels of innovation?
Come on, in this market, who's going to fund? We're going to go down that path. We're going to get to know Exsilio in detail, but we're not going to do it right now, folks. We're going to do it in part 2 of this conversation that I am hereby committing Dr. Tal Zaks to
Sorry,
do.
You're going to have to come back to tell some more tall tales.
With pleasure, colleagues. With pleasure, my
And we
friends.
will have popcorn for that one.
We will have popcorn for that one.
We will have
Absolutely.
popcorn for that one.
I will bring the popcorn. But for now, we are going to let you off the hook to meet your next commitment. And I will be in touch with your people to get part 2 scheduled very, very quickly, because I'd like it to run right on the heels of this one. This has been a fascinating conversation, and I appreciate your time. Stay tuned.
I look forward to it. Thank you so much.